Hi everyone. Today we are going to study about bacterial cell. What is the structure of bacterial cell and the different components present in the cell structure of bacteria. Bacterial cell is a prokaryotic cell. What are prokaryotic cells? Prokaryotic cells are the cells that lacks a distinct nucleus. Means they don't contain a nucleus with nuclear membrane, and also they don't have any membrane bound organelles. Okay, they are single celled organisms. Examples of prokaryotic cells are bacteria, archaea, blue green algae. Okay, so prokaryotic cells have a very simple structure. Now look at the structure of bacterial cell. So the outer membrane, this is the outer boundary. It is the outer boundary of the bacterial cell. Okay, this is the cell envelope which creates the outer boundary. Cell envelope is a chemically layered complex structure that surrounds and protects the prokaryotic cell. So this outer layer, outermost is glycocalyx. Okay, so this glycocalyx is composed of polysaccharides, and it provides protection. Okay, so it protects the entire cell. It shields the cell membrane, and also it helps in cell adhesion. Okay, this glycocalyx layer can exist in two forms. First is capsule, and another is slime layer. Okay, so capsule is a well organized, tightly bound, and tough layer of glycocalyx, while slime layer is a unorganized and loose layer of glycocalyx. Okay, so this capsule is thicker and rigid than slime layer. Now, what are the functions of the capsule and slime layer? They provide protection and adhesion to the cell. How did they provide protection? Uh, as I told you, the capsule has a very dense structure, and it prevents the penetration of antimicrobial agent and provides protection. How did they provide adhesion and why it is required? Um, because of adhesion, bacteria form a biofilm. Okay, they form a biofilm. This biofilm provides bacteria protection against antibiotic, environmental stress, etc. Also, through adhesion, they access nutrients. How? There are some bacteria which are present in our gut. Okay, they form a adhesion. They attached to the gut and access the nutrients from there. So bacteria uh, access the nutrients from their host also. Okay, now let's talk about the cell wall. This is the cell wall. Okay, cell wall contains a coating of peptidoglycan. Okay, this peptidoglycan provides strength and rigidity. So this cell wall provides structural support and protection to the cell. Now the third one is plasma membrane or cell membrane. Okay, it is a phospholipid bilayer. Okay, it is a phospholipid bilayer that encloses cytoplasm. This is all cytoplasm. After this plasma membrane, this is all filled with cytoplasm. Okay, so plasma membrane is a phospholipid bilayer. It allows the entry and exit of substances into the cell, and it is semi-permeable in nature. This is very important, very important to note that plasma membrane is semi-permeable in nature. Okay, because it allows the entry and exit of some particular substances into the cell. Okay, so after that cytoplasm, you can see the cytoplasm, this yellow colored portion, this is all cytoplasm enclosed. Okay, so cytoplasm is a gel-like substance that fills the cell. It contains various molecules enzymes, ribosomes, nucleic acids, etc. Okay, after this cytoplasm, you can see this nucleoid. What is the nucleoid? Actually, prokaryotic cells do not have a nucleus. Instead, they contain a region called nucleoid. And this nucleoid contains genetic material, typically a circular DNA molecule. Okay, so this is the cytoplasm that floats in, this is the nucleoid that flows in the cytoplasm. Okay, now you can see these small ribosomes. Okay, so these are a small structure, and they are very important because they synthesize protein. Okay, now discuss about the movement. Based on the movement, bacterial cells can be motile and non-motile. Motile, actually, the cells that can move, the bacterial cells that shows movement are motile, while non-motile do not show any particular movement. Okay, so for motility, for the movement, they have a structure called flagella. Okay, so let's see the flagella in diagram. Yes, this is the flagella or flagellum, a singular flagellum and plural flagella. Okay, so flagella is a long whip-like structure for cell movement. Now look at the diagram of the flagella. So it has three parts. This is filament. This is hook, and this is vessel body. Filament is the longest portion, and vessel body is a ring-like structure. It contain rings. Okay, like this. Okay, so flagella rotates like propellers, which allow the cells to swim through liquid environment. Okay, now let's talk about the another structures. Okay, so this is pilus. This one. What are pilus? Pilus are short hair-like appendages on cell surface. They helps in cell attachment. We can call it pilus or pili. Okay, for many hair-like appendages, it is pili, and for a singular, it is pilus. Okay, so they help in cell attachment. Just like this, there is another structure called fimbriae. Fimbriae are bristle-like fibers. They also helps in attaching the bacteria to surface. Okay, so by extending and retracting pili in a coordinate manner, the bacterial cell can twitch. The bacterial cell can crawl or twitch, okay, along the surface. Some bacteria also move by secreting slime. Slime is to move. smoothly across surface. Okay, the another one, yeah, this is inclusion. Okay, what is the inclusion? As you can see, this small circular structure. Actually, inclusion bodies are found in cytoplasm of bacterial cell. They can vary in size, shape, and composition. These are actually the accumulated proteins or some other substance. These are produced by bacteria and form inclusion bodies. Okay. Now you can see another small structure here. This is plasmid. Plasmid carry genes which provide antibiotic resistance, toxin production. Plasmids are actually the extra chromosomal. What do you mean by extra chromosomal? Means, uh, as I told you earlier, that bacteria bacterial cells have DNA in this nucleoid region, but this plasmid also contain the genetic material. This plasmid also contain the DNA, but it is not present inside the nucleoid region. It is not the I mean it is separated from the bacterial chromosome. So plasmid is a extra chromosomal. Okay. This is all about the bacterial cell. In our next video, we will discuss about gram positive and gram negative bacteria. Thanks for watching.